The topic is HIV-1 entry binding to co-receptor CXCR4. Before COVID, the world was facing another pandemic, and today 40 million people are still living with this virus. Human immunodeficiency virus is a retrovirus that attacks the body's immune system, and when left untreated, can lead to AIDS. Our focus is on the more dominant and pathogenic strain, HIV-1. To infect and deliver the viral genome into cells, the glycoprotein on the HIV's protein envelope attaches to the host cell through first binding to the primary CD4 receptor and then the co-receptor CXCR4. This co-receptor binding is a key step involved in viral entry.
CXCR4 and CCR5 are co-receptors required for HIV entry. They're chemokine receptors that bind to chemokine ligands that are highly expressed in both naive and memory T lymphocytes. Depending on the type of strain, that could be bind to either CXCR4 or CCR5. In order for HIV-1 to enter the cell, GP120 binds to CD4 receptor, leading to a conformational change to the protein envelope, thus exposing the binding site to allow co-receptor CXCR4 to bind to GP120. This triggers the GP41 subunit to form a hairpin structure in order to insert its N-terminus fusion peptide into the target cell's membrane. A 6-helix bundle then forms, which brings the membranes together so fusion can occur. Modulation to co-receptor binding can affect fusion activity, infectivity, and entry of HIV-1.
So researchers in 2016 had an idea to genetically engineer the CXCR4 receptor to have an inhibitory peptide on its N-terminus. This inhibitory peptide would bind to HR1 on GP41, indicated in orange on this figure to the left. And when this inhibitory peptide of CXCR4 binds to HR1 on GP41, it inhibits the formation of the 6-helix bundle, which prevents the fusion of the two membranes.
In this study, the researchers tested the C34 conjugated CXCR4 co-receptors on exotropic HXP to HIV-1. Exotropic is a strain of HIV that enters and infects the host CD4 cell by attaching only to the CXCR4 co-receptor. The viral entry of this strain was assessed under the presence of conjugated, that is, the engineered CXCR4 co-receptors. When compared to the unconjugated, that is, the normal CXCR4 co-receptors, only background levels of entry were detected with the conjugated ones. This can be seen in the bar graph here. The bar graph represents the percentage viral entry of HXP to HIV-1. And the first bar shows that there was 100% viral entry when unconjugated CXCR4 co-receptors were used. And the second bar shows that there was less than 20% viral entry when conjugated CXCR4 co-receptors were expressed. So this data shows that the normal functioning of CXCR4 co-receptor is important for the entry of HXP to HIV-1.
Co-receptor modulation has the potential to diminish HIV infection. CXCR4 antagonists like the C34 inhibitory peptide shown in the previous slide show very low levels of successful viral entry into target cell. In conclusion, CXCR4 targeting for inhibition is capable of blocking HIV-1 fusion with the target cell. However, antagonists can have adverse effects. Maintaining important functions of the co-receptor is essential for cell viability. Promisingly, agonists allow for some function to be retained while HIV-1 infection is prevented. Further investigation into CXCR4 agonists can give light to potential HIV therapies. This is a list of all the references used, and these are all the contributions provided by each individual.